Thank you, Professor Fiedler. I have a special pleasure to introduce the last two speakers because they have been students in the lab of Isaac Witz, and today they are pursuing their own career here at Tel Aviv University. First speaker is Professor Adit Baruch, who will tell us about the inflammatory microenvironment.
Okay, do you hear me? As a past PhD student of Isaac, it's really very exciting to stand here today and to congratulate you, Isaac, on the event of your 80th birthday and to wish you really all the best, very good health, and that you will be very productive as you are now, and that you will have very interesting and pioneering achievements in the future as you had in the past. I would like to go on to my own presentation. Okay, you will excuse me because the lights are in my eyes. So what I would like to do is to tell you about the studies on the inflammatory microenvironment in breast cancer. And I would like to show you just a few of our results concerning the mechanisms and therapeutic implications of the inflammatory microenvironment in this disease. So as many of you know, breast cancer is the most common malignancy in women worldwide. The disease usually begins in the epithelial cells of the ducts. When the malignant cells are still confined into the ducts, this is the phase of disease that we call ductal carcinoma in situ, or DCIS. But in the majority of patients, the tumor cells invade into the stroma of the breast. In many of the cases, they are also metastasizing to remote organs. And this is what we call invasive ductal carcinoma, or IDC. We actually know that there are several subtypes of breast cancer: luminal, HER2-positive, triple-negative, and other types. And you can see here that about 60% of the patients are diagnosed with luminal breast tumors. Luminal breast tumors are hormone-dependent tumors that have relatively favorable prognosis, but nevertheless, in many of the patients, we see metastasis. Like all tumors, breast cancer also has a tumor microenvironment. And in the case of breast cancer, it has a most substantial inflammatory nature. And actually, the tumor uses for its own benefit inflammatory cells and inflammatory factors like cytokines and chemokines that together act to promote angiogenesis, tumor growth, and the metastatic spread of the tumor cells. So in the immune context, the inflammatory cytokines and chemokines act in physiological conditions of mediating in a coordinated manner the infiltration of leukocytes from the circulation into inflamed tissues. But in malignancy, and this includes also breast cancer, we see that inflammatory cytokines and chemokines are very often tumor-promoting agents. In breast cancer specifically, these factors are highly expressed at the tumor site, primarily by the tumor cells, and they play causative roles in disease progression. And this means that when these factors are downregulated, what we see is reduced size of tumor growth and metastasis. I would like to address today two inflammatory chemokines. These are CCL2 and 6CL8, known also as interleukin-8. These two chemokines are known to change the leukocyte balance at the tumor site towards inflammatory tumor-promoting myeloid infiltrates. They are highly angiogenic and they promote tumor cell proliferation and migration. In parallel, I would like to address the inflammatory cytokines interleukin-1 beta, IL-1 beta, and TNF-alpha. And I would like to focus mostly on TNF-alpha. TNF-alpha stands for tumor necrosis factor alpha. And as the name indicates, if we take TNF-alpha and inject it in high concentrations directly into tumors, TNF-alpha can indeed kill some of the tumor cells. And we see this mainly happening in sarcomas, but usually TNF-alpha cannot inhibit the metastatic process. But there is a flip side to the coin of TNF-alpha, and this is that when TNF-alpha is present along the life of the tumor from development to progression at relatively low concentrations in a chronic manner, it turns to be a potent tumor-promoting factor. So here you can see, for example, an analysis that we have done in breast cancer patients, and you can see that the tumors are enriched with TNF-alpha produced mainly by the tumor cells. You can see here a biopsy section of a patient diagnosed with IDC with relapse. We can hardly detect the expression of the cytokine in healthy breast epithelial cells. And you can see that the incidence of patients who express TNF-alpha at the tissue of the breast is gradually increasing when disease progresses. And it reaches about 90% of the patients in patients diagnosed with IDC with relapse. So we were interested to know how does TNF-alpha impact the tumor cells. And specifically, we wanted to know how does TNF-alpha impact luminal breast tumor cells, because as I mentioned, this is the majority of breast cancer patients. So I would like to show you only some of the results that we have obtained here. These results were already published. You can see here the impact of TNF-alpha, only 3-day stimulation in culture on luminal breast tumor cells. You can see that TNF-alpha has induced cellular protrusions in the cells and also epithelial to mesenchymal transition. And this has led to very high invasion profile of the tumor cells. However, we know that inflammatory factors such as TNF-alpha are not acting alone at the tumor microenvironment. And we were interested in simulating the joint activities of microenvironmental factors that act in luminal breast tumors. And we decided to join 3 forces. One of them is TNF-alpha, which represents the inflammatory arm. The second one is estrogen, which is very important in luminal breast tumors, a hormone. And EGF, a growth-stimulating representative of this arm. I would like to indicate that luminal breast tumors do not overexpress HER2, one of the signaling receptors to EGF, but luminal breast tumor cells do respond to EGF. So we joined all of these 3 factors together in a treatment that we call TNF-alpha plus, and we found that in all tests that we have done, TNF-alpha plus was much more effective than TNF-alpha alone in inducing potent cell remodeling and EMT in luminal breast tumor cells. And I would like to show you only one example. This is at the level of morphology and cytoskeleton rearrangement. Here you can see the non-stimulated cells, and you can see what happens when we stimulate the cells for 3 days by TNF-alpha, EGF, and estrogen. You can see that TNF-alpha has induced the spreading of the cells. EGF also had some effect. Estrogen did not. And when we combined double combinations of the 3 factors, we could see sometimes an increased impact. But when we joined all 3 of them together, this is what we got. We got very extensive spreading of the tumor cells. This is the same magnification in all pictures. We got huge cells, very much spread, and expressing paxillin and cell proximities. Paxillin is required for increased cell adhesion and spreading. Furthermore, we analyzed the tumor cells in 3D structures, and the tumor cells here are expressing cherry. They are red, and you can see the very nice and round morphology of the non-stimulated cells, but the cells that have been exposed to TNF-alpha plus form very definite cellular protrusions that are coming out of the cells. So we found that TNF-alpha plus gave rise to cell populations with high EMT and spreading and adhesive properties. But obviously, we were interested to know what are the effects of TNF-alpha plus on the formation of primary tumors and metastases. So what you can see here is that we stimulated the cells for only 3 days in culture by TNF-alpha plus, and we got smaller tumors in cells that were stimulated by TNF-alpha plus compared to non-stimulated cells. And this is because the stimulation by TNF-alpha plus included TNF-alpha, and very much along with its name, TNF-alpha was cytotoxic to some of the tumor cells. But obviously, when we injected the cells, we injected them in equal cell numbers of alive cells. So we think that TNF-alpha has marked some of the cells to die, and they died in the mice, and this is why we see smaller tumors in cells that were treated by TNF-alpha plus. So what we see is that TNF-alpha is cytotoxic to some of the tumor cells, leading to smaller tumors, but we also found the tumor cells that resisted this selective pressure expressed higher metastasizing abilities, and you can see this very clearly here. You can see metastases The cells that were not stimulated do not form any macrometastasis, but the cells that were stimulated by TNF-alpha plus for only 3 days in culture became metastatic in vivo. We could see metastasis in about 40% of the animals. You can see here the metastasis. Again, the cells are expressing cherries, so we see them in purple, colon metastasis, liver metastasis, and micrometastasis in the lungs. So the cells that we were working with were MCF7 cells. Those of you who work with MCF7 cells know that these are very unmetastatic cells, and indeed you can see that the non-stimulated cells did not form macrometastasis. So what we see here is the impact of the microenvironment. Only 3 days of stimulation in culture leads to a very significant metastatic process. So we think that at the tumor microenvironment, when luminal breast tumors are exposed to these 3 factors that are expressed at the tumor site, TNF-alpha, estrogen, and EGF, some of the tumor cells die, but others go undergo EMT. They migrate and invade and eventually also form metastasis. So we think that while having an anti-tumor cytotoxic benefit, the joint activities of TNF-alpha with other tumor-promoting factors selects for highly metastatic cells. We then turn to ask what are the impacts of TNF-alpha on adjacent non-transformed breast epithelial cells. And the reason for asking this question is because about 90% of the patients, as I've shown you, express TNF-alpha at the tumor site, derived from the tumor cells mainly. So the non-transformed cells that are adjacent to the tumor cells are constantly exposed to TNF-alpha, and actually they are also constantly exposed to IL-1 beta, a very highly potent pro-cancerous inflammatory cytokine in breast cancer. And here again you can see that IL-1 beta is also very highly expressed at the tumor site in about 90% of the patients with relapsed disease. So we chose to stimulate the non-transformed breast epithelial cells by a combination of TNF-alpha and IL-1 beta. We found that they induce EMT in the non-transformed breast epithelial cells. These results were also published, so I will show you just a few of the results. We found that this is a slow progressing process that is completed when the cytokines potentiate the activities of the EMT regulators ZEB1 and SNAIL, leading to increased invasion of the non-transformed cells. And here you can see the combined force of IL-1 beta and TNF-alpha together, leading to much increased invasion. And another analysis that we have done was to look at acini. Acini recapitulate the ability of non-transformed breast epithelial cells they recapitulate normal breast structures. And here you can see the non-transformed cells expressing cherry. They are forming nice and round acini. And what you can see here is the acini after 3 weeks of stimulation by TNF-alpha and IL-1 beta. And you can see here 2 things. One of them is that the non-transformed cells that we found undergo EMT and increased invasion, they migrate out of the acini. And the second thing is that the acini structures collapse. So we see that the 2 cytokines together lead to collapse of normal breast structures and spreading of non-transformed cells out of the organized acini structures. So based on these results and additional findings that we had, we think that at the tumor site, when TNF-alpha and IL-1 beta are acting together, the non-transformed breast epithelial cells undergo EMT, and what we think might be happening is that they can migrate at the tumor site and reseed themselves again at the primary tumor site. And if these cells are then exposed to transformation-inducing events like nitric oxide, which is very prevalent at the tumor microenvironment of breast tumors, this might lead eventually to transformation and the formation of a secondary tumor at the primary tumor site and the formation of disease recurrence. So until now, I've shown you that by acting on breast tumor cells and on adjacent non-transformed breast epithelial cells, TNF-alpha may induce metastasis and disease recurrence. We then turn to ask, how about stromal cells? The tumor microenvironment includes also stromal cells. So what are the impacts of TNF-alpha on these cells? And what I would like to show you here are non-published results. So we decided to focus on mesenchymal stem cells that are prevalent at the tumor site. These cells are known to promote tumor aggressiveness by secreting growth factors, protumoral cytokines, and angiogenic factors, and they can also differentiate to tumor-promoting cancer-associated fibroblasts. The main source for the MSCs at the tumor site is in the bone marrow, and what we have done was to use normal human bone marrow-derived MSCs, expose them to TNF-alpha, and we have done messenger RNA profiling. You could see downregulation of 33 genes and upregulation of 131 genes in different kinetics. Many of the genes had a phenotype of inflammatory genes, androgenic genes, and genes with other potential protumoral activities. I would like to focus on the inflammatory genes and specifically on CCL2 and 6C8 that I mentioned at the beginning of my talk, and you can see the TNF-alpha increased in the MSCs, not only the messenger RNA expression of CCL2 and 6C8, but also the expression at the protein level, and these two chemokines that are protumorogenic in breast cancer were secreted by the stimulated MSCs. TNF-alpha has also induced NF-kappa B activation. We can see here the phosphorylation of P65, and when we downregulated P65 by siRNA, we could see very significant downregulation in the release of CCL2 and 6C8, indicating that TNF-alpha induces protumoral chemokines in human MSCs through the NF-kappa B pathway. Another thing that we saw was at the level of migration of the tumor cells. Here I would like to go back to what I've described previously, and this is the fact that TNF-alpha alone can induce the migration of breast tumor cells. So what we have done was to stimulate MSCs with TNF-alpha, collect the conditioned medium of these cells, and put the conditioned medium of breast tumor cells and measure the migration. What you can see here is that the conditioned medium of TNF-alpha stimulated MSCs put on the tumor cells further pushes the migratory processes in these cells more than TNF-alpha in a significant manner. So this tells us that MSCs stimulated by TNF-alpha release factors that promote migratory processes in breast tumor cells and indicates that there are crosstalks between the MSCs and the tumor cells. So we turn to look at this crosstalk and how is it affected by TNF-alpha. So what you can see here is, first of all, as I've shown you previously, the TNF-alpha promotes the release of CCL2 and 6C8 by the MSCs. It does the same on the tumor cells, and what you can see here are co-cultures between the tumor cells and the MSCs. So first of all, the co-culture itself promotes the release of CCL2 and 6C8. We don't know yet which of the cells is affected. It's possible that both of them. But you can also see here that TNF-alpha further pushes this process forward. You can see very high increase in the release of CCL2 and 6C8 in co-cultures of tumor cells and MSCs that was stimulated by TNF-alpha. So we looked whether this process is mediated by soluble factors or by direct contacts, and we think that both are involved. Here you can see the impact of TNF-alpha in trans wells that do not allow the cells to be in contact, and in contact, and you can see that the process is potentiated by the contact itself, indicating that TNF-alpha and its amplified activities required partly tumor MSC interactions. And indeed, when we use tumor cells labeled in red, they express cherry, and the MSC is expressing GFP, we can see very intimate contacts formed between the tumor cells and the MSCs. And furthermore, I would like to show you two examples. One is this, and this is the other, in which we can find yellow cells in these co-cultures. These yellow cells, when we look more closely, actually express two nuclei, and we are now checking the possibility that two nuclei hybrids are formed between the tumor cells and the MSCs. So to conclude this part, we think that at the tumor microenvironment, the interactions between the MSCs and the tumor cells further promote tumor activities. These are potentiated by TNF-alpha, leading to increased inflammatory chemokines and possibly also other protumoral factors, and lead to many tumor-promoting activities, including the migration of the tumor cells, as alluded by the studies that we have done with conditioned mediums of the tumor cells. So to wrap up, I would like to say that we think that TNF-alpha joins forces with other microenvironmental factors and promotes pro-tumoral functions in breast tumor cells, in non-transformed breast epithelial cells, and in stromal cells, leading to increased angiogenesis that I did not have time to show you, tumor recurrence, and metastasis. And here I come to the possible clinical implications of these findings. And this is the fact that until not long ago, it was suggested to treat breast cancer patients and other patients with TNF-alpha as a cytotoxic cytokine that might kill the cells. We think that this might be devastating because this might select for very aggressive tumor cells. And we think that the option for clinical implications might be different, and this is to use anti-TNF-alpha modalities to treat breast cancer patients. And we think that this is very possible because therapies against TNF-alpha are very successful already in clinical use in patients that are suffering from inflammatory diseases like rheumatoid arthritis. So at the end of my talk, I would like to thank the current members of my lab. They are excellent. I did not have time to show you all of the results and all of the projects. I would just like to indicate that Paulina was doing the analysis of TNF-alpha plus on the tumor cells. Tal was doing the analysis of the effects of IL-1 and TNF-alpha on the non-transformed cells. Shalom and Yulia were working on the MSCs. Tzipi contributed to all of these studies. I would like to thank our collaborators in Tel Aviv, Israel, and abroad. And at the end of my talk, I would like really to deliver, Itzik, to you a very warm bouquet of flowers and to thank you. And there are two reasons. One of them is that you introduced the field of tumor microenvironment to me, and I decided to follow it because I thought it is very exciting. And the second reason is because I learned the strength and beauty of immunology because of your lessons when I was at the third year of my undergraduate studies. And this is where I remained. When I first registered to the life sciences faculty in Tel Aviv University, my intention was to study zoology and nature preservation. And then I took the immunology course that was given then by Asha Franzdorf, late Asha Franzdorf, and Isaac. And after the fourth or fifth lecture, I decided that I convert into an immunologist, so I thank you.